going to move to the Y chromosome polymorphisms. Sofie Claerhout will talk about a panel-based MPS approach.
Hi everyone, my name is Sofie Claerhout and I'm a PhD student of Professor De Korte in the Laboratory of Forensic Genetics in Leuven, Belgium. And today I will talk about our custom-made massive parallel sequencing panel, what we call the CSYseq, which targets a lot of interesting Y chromosome markers on the very promising Y chromosome. People always ask me, why are you so interested in the Y chromosome as you don't even have one yourself? But I'm sure most of you know that it's because 95% of it lacks recombination with the X chromosome, meaning that it is inherited from father to son in a relative conserved manner. Therefore, biological kinships can be identified using Y chromosome polymorphisms, such as the slow mutating SNPs and the fast mutating YSTRs. This gives us several advantages for forensic DNA analysis. For example, we can identify the male-specific part in mixed male-female samples on the crime scene. Using SNPs, we can have a lot of biogeographical background information, which is valuable for forensic investigators. And typing YSTRs, we can identify paternal lineages, useful for paternity testing, DVI, and familial searching, whereby investigators are searching for the relatives of an unknown perpetrator. Until now, the golden standard to genotype these markers still mostly relies on capillary electrophoresis, so fragment analysis. As this method has its limitations, more interest has gained toward new techniques such as massive parallel sequencing. This gives us several advantages. For example, we can target more markers at once, so we can combine STRs with SNPs. We can multiplex samples together, and of course, MPS will gain single base information. As MPS has already gained some interest today, this led to the development of several sequencing panels. And here listed, close to my hand right now, are the sequencing panels who contain Y chromosome polymorphisms. As you can see, only the latter one, where I'm pointing to, contains a high amount of Y SNPs, but none of these kits combine STRs with SNPs. So therefore, our goal was to create a panel, an MPS panel, Y chromosome specific, targeting an enormous amount of STRs and SNPs. In order to create this panel, our CSYseq, we selected regions of interest according to literature. For the YSTRs, we based our selection on Y chromosome position, repeat motif type, and, if available, the mutation rates. For the Y SNPs, we based our selection on the minimal reference Y tree, which includes the most important branch-defining SNPs to target the whole phylogenetic tree. Primer pairs were designed using Design Studio by Illumina, and we prepped our samples using the TrueSeq custom amplicon low input kit of Illumina, to then further sequence them on the MySeq 2x300 base pairs, which is paired-end sequencing. Then samples were demultiplexed, and variant calling was mainly done through StraightRacer for YSTRs and through Y-Leaf software for Y-SNPs and haplogrouping. So Illumina gave us a list of regions that our panel would target in theory, which we then compared to what our panel targets in practice, so after data analysis. So in theory, according to Illumina, our panel targets 857 amplicons, which is very nice, and they have an average length of 250 base pairs, all distributed over the euchromatic region of the Y chromosome. These amplicons, in theory, target 227 YSTRs and more than 12,000 Y SNPs, according to the ISOC-Y-Browse database. In order to verify these amplicons and these markers, we already sequenced one female sample, not mine, and 130 male samples. And these Males are also genealogical pairs, so they are biologically related with a few generations apart. So our samples cover in total over 1,000 generations until now. So after data analysis using straight tracer, we analyzed our 227 YSTRs, and until now, as data analysis is still ongoing, we already had 120 well-sequenced YSTRs. So that means ease of interpretation, highly Y chromosome specific, and a high depth of coverage. Almost 40 YSTRs were badly sequenced and contained a low depth of coverage. 30 YSTRs exhibited extreme overlap with the X chromosome and or autosomal chromosomes. And as one of our goals is to mainly target the Y chromosome, we further excluded these ones. And then the latter group are the incorrectly typed YSTRs by straight tracer and the complex repeats, so double repeats, multi-copies, wherefore separate loci could not be determined. However, for the latter group, we are still trying to increase our well-sequenced YSTRs using other programs and other settings. But for these 120 well-typed YSTRs, we already gained some information about these markers, about the allele calls, the allele frequencies, the allele ranges, through the non-related males. By comparing the Y chromosomes of the genealogical pairs, we could identify their mutation rates, and 42 of them, which are not on this slide, did not have any allele change, and 13 of them were identified as rapidly mutating YSTRs. And we blame the length of repeat motif and the allele size as an influencing molecular factor. So this for the YSTRs. Then on to the SNPs. After data analysis, we targeted over 15,000 Y SNPs, which is a huge amount. And they can be distributed into 3 groups. We have the private SNPs, we have the SNPs with unknown phylogenetic position, and the majority belongs to the haplogroup-specific SNPs. And these cover more than 1,400 deep subhaplogroups, all distributed across the phylogenetic tree, so covering the main haplogroups from A to T. This means that any male with any biogeographical background would, in theory, be possible to haplogroup with our panel. If we translate this to our samples living in Belgium and the Netherlands, then we see that for all subhaplogroups in our population, a lot of SNPs were typed, and here you see that they are divided into the ancestral typed SNPs and the derived typed SNPs. If we then translate this to the success rate of our panel, so the massive parallel sequencing, then we see that 66% of our samples were better typed using massive parallel sequencing in comparison with snapshot technique used in our lab. On average, 4 branches deeper within our samples were typed using massive parallel sequencing, and we saw a maximum of 11 branches deeper. So that is quite nice because snapshot technique is sometimes very time-consuming. Then on to my conclusion. Now we have this CSYseq targeting a lot of interesting Y chromosome markers, so a huge amount of STRs and SNPs, and data analysis is still ongoing. But for now, our panel has a high Y chromosome specificity, so that means ease of interpretation for mixed female-male DNA samples. We have a high discrimination power according to the different mutation rates to discriminate between related and non-related males. And this all together in one panel, in one assay, and in one protocol, getting as much as information as fast as possible. This being said, I would like to thank everyone making it possible for me to do research on this very promising Y chromosome, especially my colleagues in the lab. And of course, I would love to thank you for having me and for your attention. Thank you very much.
Thank you very much, Sophie, for the nice presentation. And I think that we are having questions this time, so it seems that you provocate the audience. And starting for the first one, would it be practical to leave in the overlapping 24 autosomal for possible concordance purposes?
Oh.
Okay, it's not there. Do
So
you need to repeat?
yes, maybe.
So asking if it will be practical to leave the overlapping 24 autosomal for possible concordance purposes.
So it would be practical, and indeed we know which ones are overlapping with the autosomal and the X chromosomes. So we're not sure yet if we will leave them out. So if we will ask or develop a new panel to leave these amplicons out, because it's also nice to have more information. But mainly we will focus on the Y STRs who will target the Y chromosome, just for our main goal to make it and maintain it Y chromosome specific.
And another question, did you also compare closely related males, let's say father-son pairs or brothers, to see if they could be differentiated?
No. We would love to, but according to our ethical commission, we are prohibited to sequence closely related individuals like brothers and father and son, just because I don't think we want to start, because actually the thing is our samples were sampled for the investigation on extra pair paternity. So we had our minimum generation level of 7 generations, because when we incorporate brothers or father and son and then we would find extra pair paternity, then maybe some kind of problems in the family or something.
I think we are a little bit over the time, but I'm going to ask another one because I see that you have here many votes for another question. What kind of sensitivity levels did you reach with this essay?
So the thing is, we had this on the MySeq done. So if in terms of sensitivity you mean like the amount of reads, then the MySeq paired-end sequencing gives us 50 million reads per run. And when you then want to sample 96 samples, then you have some kind of reads coverage of 500,000 per sample. And if you then divide it by 1,000 for the amplicons, then we have like 1,000 reads per amplicon for this panel. Yeah.
So we have to move on. Thank you very much.
Okay, thank you.